Okay. Let's see. Training human neurons. Okay, yeah, looks like the main principle is neurons want to decrease their well, it's based on the free energy principle. So neurons wants to make sure they can predict future stimulus. And the best way to do that is apparently to both make better observations and to sort of basically do actions. I believe how they do it is they have inputs as electrical signals to the neurons, and then outputs as electrical signals out. And if the outputs are the desired output, they give the neuron something very predictable. So obviously the neurons would want to prefer that. And then if the output is not desired, right, so like a invalid control or a control that makes the sort of the make the player lose the game, then a random signal that cannot be predicted will be the feedback. So the neurons learn to give the correct outputs. It doesn't seem like the most efficient version of applying free energy principle, but it's very interesting, and I'm trying to look for what are some of the finer details about how they accomplish this. Okay, this is cell biology. I am not too familiar with this. Okay. Input sensory information. Let's see. Ball position of, okay. Moving the paddle. Interesting. Hit or miss. Okay. Process. Okay, so the process is like the output of the neurons, and the input is some electrical stimulus, I'm guessing. It could be anything. Any electrical stimulus, as long as it reflects the position of the ball. Right, XY coordinates would be fine, I guess. So ball, okay, so this is missing the ball, unpredictable feedback. And then if you hit the ball, it's a predictable sensory feedback. Okay, but like, what about all the times when the ball is traveling? Okay. What's this silent feedback thing? Stim SIL. Okay, so this is basically different feedback configurations, right? Open loop feedback. Okay, I guess no feedback, ball continues. Huh. Let's see what the description is. Different dish brain environments are created by altering the pattern of sensory information. Okay. Oh, I see. Sensory information is what you get when you're not in a reward state, or where you're not in one of the two states where you've successfully catched the ball or missed the ball. Feedback. Colored bolts, okay, or no stimulus, red crosses. To demonstrate low latency closed loop feedback system and the silent treatment. So silent treatment is basically the same. Oh, like basically there's no feedback to tell you about, like, so basically this is like giving both sides the same feedback, being no feedback. Okay. Oh, I guess the only differentiating factor is that in the case where you miss the ball, I guess the ball resets to some central location. Okay, yeah, so normal pong game, the ball resets to the center. Low latency closed loop feedback system, okay. I guess this is still closed loop, the second one's still closed loop. Because you do get some feedback, it's just not, like colored feedback, I suppose. No feedback system. Demonstrate an open loop feedback configuration. Okay, but like what kind of feedback are you even getting? There's not even a special state telling you that the ball has bounced. No sensory information. Okay. Interesting. So where are your methods here? Why is it just, okay, results. Okay, you grow some cells. Oh, I guess it's rat brains. Okay. That's a good correction to make. Training rat neurons to play pong. Okay. Long-term cortical neurons that form dense connections. What are cortical neurons? Cortical neurons are the ones in the cerebral cortex. Okay. So not in the brainstem or anything like that. Huh. Interesting. Oh, these rat neurons could be maintained for months. Interesting. And they can even differentiate. Direct reprogramming, okay. This is just confirming the like that these neuron cells are in fact normal, I suppose, and not dead or diseased. Building modular real-time platform. Okay, right. This is important. Okay. The vendor max 1 soft. Okay. So there's a real-time system that these neurons are placed on. Okay, so sensory information is just electrical signals stimulation at a similar level to action potentials in a neuron. Right, so it doesn't like electrocute the neurons essentially, right? Using coding schemes described in STAR methods. Okay, there's only 3 distinct informational categories. Predictable, random, and sensory. Okay. Neuron culture. Okay, write sensory data to neuron culture in a closed loop system so that neural action influences incoming sim. Right, okay, just parts of the neurons are stimulated, right, I'm sure. What are these nice graphs? Cells display spontaneous electrical physiological activity. Okay, but that's without playing the game, right? Very interesting, I guess. Electrodes have a coarse yet topographically consistent place coding. Okay. Let's see. So the activity of the motor regions, which is some arbitrary region, that activity is used to move a paddle. If it did not result in an interaction of the ball by the paddle, so if it did not hit the ball, unpredictable stimulus was delivered. Okay, for 4 seconds. Okay, wow. After which time, so to the sensory neurons, I'm guessing. After which time the ball stimulation would recommence on a random vector. Okay, but like, In contrast, if a successful interruption occurred, a predictable stimulus was delivered across all electrodes simultaneously. Okay, so I guess all of this like stimulus was simultaneously delivered. Interesting. Briefly interrupting the regular sensory stimulation. Okay, before the game continued predictably. Okay. That's very different. Primarily, okay, why 4 seconds? That's a long time. And where? Why is this so unspecific? Different motor region configurations to verify. Okay. Sure, okay. Distinct areas were defined as motor regions, okay. Where activity motor region action 1. Oh, I see, moved paddle up. Okay, so this is up and down is specified by 2 different regions of neurons. And it's not a continuous thing, it's just 2 discrete choices. Okay. This fixed layout means that monolayers of cells is a random distribution that is arbitrary in relation to the motor configuration. Okay. Need to adopt distinct firing patterns through self-organization. Okay. Sure. That's reasonable. I don't know what they mean by one layer though. Oh, I guess they don't specify one layer. Could be multiple layers. Alright. 4 hertz, so 4 stimulations per second. That coded the location of the ball, I guess. Oh, the location of the stimulation corresponds to the position of the ball. Interesting. On the y-axis. Oh, so it doesn't even tell you like how close the ball is to the paddle, just like the position of the ball, how far up and down it is. Yeah, I guess if you constantly just track that using the paddle, this works. Okay. Wait, how long did they train for? That's an interesting question. Okay, pilot study 2. Wait, what? Where was this? Okay, pilot study 2, investigate different configurations, introduce activity-based weighting to account for cell density or active. What? What does that mean? Account for cell density or activity differences. So I guess this is more of like a hardware issue that they're addressing. Okay, if you could change the combined rate. Place coding, method of data input. Rate, what does that mean? What is the rate here? This combined rate and place coding has complex biological similarities, conceptually compelling biological similarities to the rodent barrel cortex. Okay. Okay, I guess this is something I won't understand because I don't have an understanding of rodent biology. Gameplay for the final 15 minutes of each culture type was compared. Okay. Okay, I guess the final pilot study is really much more effective. Oh, interesting. So human cells are also tested. Okay. Interesting. And they're more effective. That kind of makes sense. Increasing the amount of sensory information improve performance. So like more training data is better, I guess. That's nice. Is this figure 4? Yeah. Neurons or mouse neurons. Okay, put it on a chip. Okay, this interfaces with Pong. Electrode row. What is this? Motor region action 1, motor region action 2. Okay, there's 2 of them. Interesting. And there's a sensory area. Okay, but the and the sensory area, I suppose, is tracking the Y position of the ball. Okay. And there's a bunch of stimulation electrodes. Okay. That kind of makes sense, I suppose. What is this? Maxwell's alpha Walker process recording spec. Oh, this is like a systems diagram for the electrical interface. Okay. Average rally length. Human brain cell. Okay, so I guess, yeah, for both configurations, the newly added stimulation patterns helped a lot, especially for human cells, I suppose. Okay. What is this? Distance to paddle, distance to wall. Okay, what is this telling us here? I mean, you would want distance to paddle to be as small as possible. This is kind of hard to read because you have to line these both up. Distance, like, you want the paddle to be near the ball when distance to ball is low. I think it's like maybe sort of happening. So yeah, this is kind of unclear. Sensory predictable summed activity. Yeah, I guess activity would be highest when all cells are receiving stimulation. Yeah, activity also really rallied near when they successfully hit, and I guess forgot to rally whenever they didn't hit. That's kind of interesting. See, all these rallied prior to hitting. Oh, except maybe this one. Maybe this one just got lucky. Yeah, for this one it did not go up fast enough. Did not rally. Interesting. Okay. Yeah, except, well, how effective is this? Do we have a graph for like training time? Train induced. Okay, but like I want to know what the training time for this is. What is this? Very substantially over time. Okay. Oh, hold on. What's this? A linear regression of the correlation to found to decrease its time significantly until approximately 5 minutes of the gameplay. After this point, we're trained in 5 minutes. Okay, this is the job, I think, at this point for some automated reading software. Because, I mean, this is kind of ridiculous. Compare the sample efficiency of these networks compared to gradient descent. Like, I suspect, well, okay, we know that the sample efficiency of neurons in human brains are at least, several orders of magnitude higher than that of artificial neurons trained using gradient descent. But is this true, just trivially through these like very basic training approaches, right? And I mean, this is a very basic approach that just penalizes you whenever, basically gives you a bit of a, not even a shock really, just a bit of a unpredictable like stimulus whenever it does something wrong. So yeah, it's not clear to me if this sort of training regime will be superior in terms of sample efficiency compared to gradient descent. But it would be very interesting if it is, because it would basically show that, neurons in general are much more sample efficient. Okay. Does not provide explicit quantitative comparisons. What? This is ridiculous. Okay, we're just gonna have to copy all the words on this PDF. There's no way it doesn't ever talk about there's no way that it does not talk about the training efficiency, right? I mean, that would be such like a that would be like a huge part of this that would be interested in knowing about. Kind of hard to excuse not talking about it. Like they're trying to hide. Obviously they're not trying to hide something, but I mean, they're probably not trying to hide anything. Maybe it took them like an extremely long time to train these neurons to play Pong, and it failed multiple times, right? Even like their eventual process, their final process might have had maybe like a 1% or 10% success rate. That could be something that would cause them to try to hide this information, but it doesn't seem like that would be the case. Reputable journal and all that, and a lot of media attention for this as well, although that's never guaranteed that the results are actually good, just that they're sensational. Learning was observed within 5 minutes of gameplay. Really? Applying implications from theory actions, we find apparent learning within 5 minutes of real-time gameplay, not observed in control conditions. Okay, but how good was the gameplay? Hold on, okay. Figure 5b. 5b. Let's just search 5b here. Oh, 5b right here. Okay, average rally length. Nani? At around 0 to 5 minutes. Okay, wait, what is this group? This is well, the rally length is like 1 or 2, which means they're basically missing almost every shot. So this is not great, I think. And what is the what is the correct, like what is the optimal sort of group here? What is this initial feedback? What is this? Remember they talked about it somewhere. Yeah, STEM. Okay, but that's not one of the groups. What? Yeah, where's the group, man? Okay. Mouse brain, is that human cortical neurons? What is RST? And IS? Control groups. What is, oh my god, these abbreviations, man. Oh, CLT, CTL, CTL. Okay, what is this? Media were compared with media-only controls. Cortical cells, okay, mouse and human cortical cells. Media-only controls. What does that mean? Red sessions, where active cultures control the paddle but receive no sensory information, and in silico controls that mimic all aspects of gameplay except for the paddle were driven by random noise. Okay, so all these controls are just doing nothing. Okay, like we expect them to have random performance, right? Okay, this is no sensory input, this is no sensory feedback, and this is no well, this is just random noise, right? Okay, at 5 minutes is actually worse than random. Okay, but then it becomes better than random at 6 to 20 minutes. Okay, but the rally length is 1. This is like really unimpressive. Oh, what's this? Long rallies. Percentage of long rallies. What is a long rally defined as? Like, I mean, over here it's saying the maximum rally was 1.5. Which is kind of embarrassingly sad. Average paddle movement pixels. Okay. I mean, you get a bit higher, maybe. Just marginally a bit higher. Okay. What is this? Percent axis. What is this? ACEs. Okay, but like, this is really sad. basically this paper has not really demonstrated much learning at all. Like, it is barely any learning. Like, it is probably like a barely measurable amount of learning. One rally, I don't like control, like random was like 75%, maybe like 80 to 90% accuracy. Well, okay, an 80 to 90% chance of, like hitting a rally, I guess. Right? With the potential of maybe hitting a rally and a half. Yeah, and then your trained model is, yeah, pretty much similar. Like maybe like a 0.2 more rallies with about just as wide of a variance. Like, this is not really. Oh, okay, what is percent long rallies? Long rallies. Okay. As aces, number of gameplays with greater than 3 consecutive hits are long rallies. Okay. Reduce the amount of they missed. Okay, but long rallies are like more than 3 hits. Okay, but more than 3 hits is, what is the percentage of that? Okay, that's still pretty low. Oh man, this is kind of like deceptive statistics. Like, more than 3 implies that there could be a lot more than 3. Except I'm betting that most of them are maybe capping at 4 or something like this. Like, average rally length, yeah, like 1. Like, this is not great. Okay, what is this? 10%. This is barely better than random. Like, you move to 9% to 10%. This is not. I don't think this is significant. Like, I mean, compared to controls. Although compared to, the original, I guess you improved by like maybe 3%. And over here by maybe 0.25 of a rally. Okay, but this is like really, maybe 0.5 of a rally. This is still really kind of sad. Okay, well, this is unfortunate. I mean, it can probably learn. It doesn't learn very well. It's kind of sad. That's okay. Like, I guess we have a better view of, I guess sort of state of the art. This is probably not state of the art anymore, but the state of things in training neurons to play video games. Essentially, mostly very ineffective, and but demonstrates at least some way of training neurons. Well, it demonstrates one of the inductive biases, potentially out of many others, that neurons use to as a signal to self organize. Right, so it will try to minimize unpredictable information, and they've leveraged that in order to basically train it to improve slightly at playing Pong. But unfortunately, this signal is like not very strong, and probably also they haven't trained for long enough. So the resulting performance of these neurons is very low when we're talking about like Pong performance. Right. I'm gonna end it now. See you guys.